Okay, hello everybody. So this is a little overview, a little review of the stuff you need to know for Lab Exam 2. So a little preface here for all the labs we've done for this practical. You need to be prepared to answer any questions that were on those labs. So on those labs usually there's a number of questions, little review questions or questions along the way. Some of those exact questions will show up on the lab practical. Now I'm going to make a separate video where I'm going to go over those labs, labs 7 through 12, so that we can, be sure about those questions. Okay, so the lab 7 was the dichotomous key lab. So if we give you a dichotomous key, and we will give you a dichotomous key, and either some kind of image of something or maybe an actual specimen, you need to identify it to family. So the key will have, will go to family, in other words. Also, you need to be able to construct your own dichotomous key. So what we might give you is a series of organisms that have varying features, kind of like those buttons. Remember the button lab, or not the button lab, but on your lab 7 there were several different kinds of buttons, and you had to generate a dichotomous key using their different features. So you need to be able to do that as well and have it be in the proper format of a dichotomous key, and using as few steps as possible. Okay, so now let's talk about Protus, Lab 8. So this is kind of a big lab. This is a really big lab. So we had several different organisms we looked at, representing the different supergroups, and usually more than one phyla within that supergroup. So you need to be able to, for all specimens we looked at in that lab, be able to identify their supergroup, identify what phyla they are in, identify the structures you were required to label on the drawing. So if you have not been doing your labeling, this is where you're going to kind of have a little bit of a problem. But that information is in your lab, so you can go and look what you were supposed to label. You also should know their mode of nutrition. In other words, are they an autotroph? Are they a heterotroph? If a heterotroph, are they a parasite? Things like that. So, and we're going to go over some of this stuff in this review. You should know a little bit about their mode of locomotion. So some of these had cilia for locomotion, some like the amoebas used pseudopodia for locomotion, some use flagella for locomotion. That's another thing I'll touch on as we go through this review, and I'll talk about these things in those lab reviews I'm going to also post a video of. Also, we had a couple of important parasitic species we talked about. So in those cases, you actually need to know the genus name, and I'm going to explicitly talk about those genera, and you need to know the disease it causes. I know, that's a lot. So let's take it step by step. So, supergroup. One of our supergroups is supergroup Excavata. There were 2 organisms in this supergroup that we looked at as examples. One is the organism Euglena. So here is a Euglena. Not readily visible here in this particular micrograph, but they typically have a flagella. Actually, they had 2 flagella, one long flagella and one shorter flagella that is usually not visible. So that is their mode of locomotion. Another thing about Euglena is many of them are what we call mixotrophs, meaning that they have some photosynthetic ability, but if sunlight is not readily available, they can also act as a heterotroph and eat food from their surrounding environment. Another member of the Excavata we looked at is this organism here, Trympanosoma. So Trympanosoma is one of our parasite genera you need to know, okay? Trympanosoma causes the disease, oh, I can't even spell this, ridiculous, sleeping sickness. So what you're actually looking at here, these are red blood cells. And then this is the protist, Trypanosoma, supergroup excavata. So another another supergroup we looked at was the supergroup Archaeplastida. We actually looked at several organisms that were from the supergroup Archaeplastida. One of them is this Spirogyra. So depending on what you're looking at, so we have the vegetative version of Spirogyra, where we have those spiral chloroplasts and the nucleus inside here. But you also should be familiar with the reproductive image of Spirogyra. So especially this structure here, this is called the conjugation tube. And this is how we're going to have the male gametes moving into fuse with the female gamete cell. And as a result, we end up with a zygote. So that's not a gigantic nucleus, that is a zygote. Also in the supergroup Archaeplastida, we looked at Volvox, which is a colonial green algae. So each of these structures here are actually made of many, many, many smaller cells, little flagellated cells that look kind of like this. And we have thousands of them making up the outer edges, sort of this hollow ball, and they sort of work together and coordinate to move around and feed. And then in the interior, we have daughter colonies forming. And when these daughter colonies are mature, they will exit out of the parent colony and become a full-fledged colony of their own. So that is an example of what we might call a colonial green algae. This would also be considered a microalgae, as would Spirogyra. This would also be considered a microalgae because of its very small size. And we would also say this is a filamentous algae. Now, we also have some algae that are truly multicellular. And this sea lettuce, Ilva, is an example of this. So this is actually a multicellular green algae. Okay, so then here's just the three of them together as a comparison. Okay. Oh, by the way, let me back up for a second. This is phylum Chlorophyta is the phylum here. And I'm going to go over all these phyla when I do the videos for the, where I go through the labs as well. So also in the Archaeplastida are the red algae, phylum Rhodophyta. So the red algae have that pigment phycourethrin, giving them that red color. Also, some algae that we call, some red algae known as the coralline red algae have calcium carbonate within their cell walls, which give them this hard, rigid structure. So that's a couple things to know about this phylum Rhodophyta. So the protists also include the supergroup SAR. This is our kind of our biggest supergroup. So one of the, or I should say a couple of the phyla within the supergroup SAR, we would also sort of generally consider algae, but they're in a different supergroup, so sometimes that's a little confusing. So what we might call the brown algae are actually supergroup SAR. Okay, so let's look at a couple of organisms within the supergroup SAR. So one of these are the diatoms. So the diatoms are small. They have what we call a test or a shell, also known as a shell, made of silica, which is a primary component in most glass. So they sort of have this outer, so they sort of appear like these little ornate glass boxes. They also have photosynthetic pigments, so they are autotrophs that can do photosynthesis. Also, within the SAR, another organism that we might call brown algae is this organism right here. We looked at a couple different brown algae. And so one of the things you need to know about this is these are the only group that are all multicellular within the protist. So this is phylum Phaeophyta. And Bacillariophyta for the diatoms. And I'll go over this again in the lab review. So this phylum Phaeophyta, all multicellular, okay? Everybody's multicellular in this. So that's one kind of key thing to know about this phylum. Another key thing to know about this phylum are that it has some structures that are sort of superficially plant-like, although they're not plants, but they have, I'll erase some of this hot mess here. They have this root-like structure called a holdfast. Again, these are not true roots. They don't have vascular tissue, and also this isn't a plant, but it would be used for attachment to some kind of substrate like a rock. We have what is called the stipe, kind of hard to see here, or the stipe, which is a stem-like structure, plays a similar role to a stem in that it serves to hold up the photosynthetic organs. And then we have the blade, which is sort of analogous to a leaf. It's thin, it's typically flat and broad, and its job is to intercept sunlight for photosynthesis. Another thing we have in these brown algae oftentimes are these gas bladders. And remember, this organism is in the water, so these gas bladders will help raise up the blades in order to intercept more sunlight for photosynthesis to occur. Okay, so something I should have mentioned a minute ago. Okay. I don't know if you can tell, but it's early when I'm recording this. SAR stands for the 3 subgroups within the supergroup. So we have the Straminopoles, and these are the Straminopoles. Right, we have the Straminopoles, we have the alveolates we'll talk about next, and we have the Rhizarians. Those are subgroup clades within the supergroup. Okay, so let's talk about the A in supergroup SAR. These are the alveolates. So these alveolates are named for the presence of alveoli, which are sac-like vesicles found beneath the plasma membrane of its members. So there were 3 main phyla within the alveolates that we looked at, starting with phylum Ciliophora. Sorry, I can't spell today. So the phylum Ciliophora includes this organism, Paramecium. So one of the things it gets its name from, or the feature does get its name from, are these, kind of hard to see, these fine projections called cilia. Other features that are really prominent in Paramecium are those really distinct macronuclei. So one of the things you need to know about Paramecium is that it has sort of an interesting reproductive cycle in that the process of actually splitting and reproducing itself is separated from the process of sexual recombination. So remember this slide? This is something called transverse fission. So this is how it's going to reproduce. This is how one paramecium will become 2 paramecium. It's going to just basically pinch in half like this. This is very much like binary fission that we see in bacteria. And it's all well and good. However, it doesn't generate any sexual recombination, so that's problematic. So the way that the paramecium generates new genetic combinations is through the process called conjugation, which is what you're seeing here. Two paramecium are going to align and swap their micronuclei. Okay, so the paramecium have the really obvious macronucleus, but they also have smaller micronuclei, which they will swap with each other in conjugation to generate genetic diversity. Okay, another ciliate that we looked at is this organism, Vorticella. So this guy's kind of cool. So it's got this stalk here that will typically attach to a substrate. And the top part here, this is where food will go in, and we've got the cilia, are less obvious than in the paramecium, but we do have cilia up here. And this stalk is able to rapidly retract if it senses some kind of threat is, imminent, like a predator or something. Okay, another important phyla, so we're switching phyla here. By the way, the Vorticella is also phylum Ciliophora. But now I want to talk about phylum Apicomplexa. So phylum Apicomplexa includes the medicinally, well, it's causes a disease, that's why it's medicinally important, includes this organism Plasmodium. So Plasmodium was the one that was the most annoying to find on the microscope, if you remember this one. This is the parasite that causes malaria. So this one's important to know the genus name up, and you need to know that it transmits malaria. So again, most of what you're seeing here are red blood cells. The actual Plasmodium is very tiny. Some of these red blood cells are infected with many, some just one, and it usually just looks like a little purple dot. It's really, really small. Of course, it doesn't actually look like a little purple dot, but with the resolution of our scopes, that's what we're able to see. So our last phylum within the alveolates that we're going to talk about is the phylum Dinozoa, aka dinoflagellates. So a couple of features on these dinoflagellates that are worth noting. So this dinoflagellate does have photosynthetic pigment, so it can do photosynthesis. Also, we have typically two flagella, one that's going to be found around the body in this flagellar groove. So you need to know this flagellar groove and that there'd be a flagella there. And then usually there's also one trailing flagella. Also, this organism is armored with cellulose plates. So the cellulose plates are just what's making up the outside and kind of give it this sort of, I don't know, kind of interesting structure here. So those are the cellulose plates on this organism. Okay, so now we're to subgroup Rhizaria, the R in SAR. So there are two phyla in this supergroup we're going to look at, phylum Radiolara and phylum Foraminifera. So just, if you just took so phylum Radiaria, phylum Foraminifera. So these guys sort of differ in the structure of their test. So radiolarans possess a single-chambered silica test, whereas the foraminiferans possess a multi-chambered calcium carbonate test. One thing they have in common is that they both contain thin, thread-like pseudopodia that are going to extend out of these tests. And these pseudopodia, so they have you can't see them, but and this is obviously not to scale at all, but they'd have these thin thread-like pseudopodia that would be able to, for example, engulf a food particle or something like that. Okay, so now we're on to the supergroup Uniconta. Sorry, I'm hoping I'm not making you seasick here. So we're going to look at a couple of different organisms here. So from the phylum Amoebozoa, we are looking at the amoeba. So this organism is heterotrophic and moves by pseudopodia. So this organism, rather than having the thread-like pseudopodia we saw in the other group, this has what we might call lobe-like pseudopodia or tube-like pseudopodia, and that's what it would use for movement. Also for feeding, it would sort of trap a food particle and engulf it using the pseudopodia to help it out. So also within this group, we have the slime molds. So there are both cellular slime molds and plasmodial slime molds. And one of the important structures to know on a slime mold is this structure right here called the sporangium. And what is going to be produced in this sporangium are spores. So these structures, these sporangium, are one of the reasons we kind of thought for a while that these slime molds were actually fungi, but they're not. They are protists, but they do produce those spores within sporangium. Okay, so the next lab I want to talk about is the fungi lab. So again, we have several phyla that we looked at. So you need to know for the fungi, the kingdom, that's easy, right? Everything is kingdom fungi, okay? If it's a fungus, it's kingdom fungi. By the way, for the protist, there is no kingdom, right? Because what we thought was kingdom protista is paraphyletic, so we use the supergroup system instead. There are also several phyla we need to keep track of. And within these phylum, in particular, we're going to pay special attention to the sexual reproductive structures that are specific to each phylum. We're also going to look at the main vegetative growth structure of a fungus, and also you need to know any structures you're required to identify on your drawings, and any questions you were asked in that lab are also fair game. Okay, so let's talk phylum Zygomycota, which gets its name from this structure, the zygospore. This is part of the sexual reproductive cycle. So if you think back to, and I would encourage you to go back maybe to the life cycle for this Zygomycota, and it had both a sexual life cycle and an asexual life cycle, and the production of these zygospores are part of their sexual life cycle. They also can produce asexual spores through these structures called sporangium. So the main body of a fungus is called a mycelium, and these mycelium are made of these thread-like hyphae. These thread-like hyphae are capable of asexually putting up a sporangia that makes spores inside, and these would be asexual spores produced by mitosis. So that's the asexual part. Great for dispersal, but you don't really get any sexual recombination that way. Okay, so let's talk about the Basidiomycota. That's another phylum we looked at. So things that you would think of as mushrooms, like mushrooms you might buy at HEB, those are phylum Basidiomycota. And again, just like all the other fungi, it's made of those thread-like hyphae, which when taken collectively, we call them mycelium. Now, One of the things about the Basidiomycota is that they have a long-lived dikaryotic stage. So what does that mean? So if we say something is oh goodness gracious, this doesn't want to work with me, heterokaryotic. It means that there are more than one genetically distinct nuclei within a cell. And if we specifically say it's dikaryotic, that means in each cell there are two nuclei and they are genetically distinct. One would be from, one mating type and one would be from the other. So in other words, plasmogamy has occurred, but chirogamy has not. And in the Basidiomycota, we have typically a long-lived dikaryotic stage where the cells of the mycelium are made up of cells that have two genetically distinct nuclei. And in the Basidiomycota, it's really only within specialized structures that we're going to have plasmogamy giving us a diploid cell. And where do we find these specialized structures? Well, first we look to the fruiting body. So most of the fungus is underground as this tangled mat of hyphae known as the mycelium. But when it's ready to reproduce sexually, it sends up a fruiting body that in the Basidiomycota is called the basidiocarp, aka a mushroom. If we were to look at the gills underneath the cap of the mushroom and zoom in, each of these gills would look something like this. Now, you see all those little purple dots? Let's zoom in on one of those little purple dots. Kind of hard to see, but that's one of them. That structure is called a basidia, or a basidium would be the singular. So basidium, or if we're talking plural, basidia. So within these structures called the basidia, the chirogamy fusion of the nucleus will occur. So what was a dikaryotic cell will fuse, the nuclei will finally fuse, so we'll have a cell that has a diploid nucleus. That diploid nucleus will divide by meiosis to produce 4 haploid cells. These haploid cells get packaged into the 4 haploid spores known as basidiospores that can be found hanging off the end of the basidium. Okay, so there's always going to be 4 basidiospores, and I know it might not always look like there's 4, but see some are falling off because it's releasing some of those spores. But that is the characteristic number of spores for phylum Basidiomycota. Now, the phylum Ascomycota has a very similar life cycle in that it has a long-lived dikaryotic stage, and it will send up a fruiting body. This fruiting body is called an ascocarp. And on this ascocarp, if you zoom in, we find specialized structures called, whoops, I cannot write today. Singular would be ascus, plural would be asci, and they get described as sac-like structures. I think they look more like finger-like structures. And it is within these asci, same deal, where chirogamy will actually occur. And so what was a dikaryotic cell becomes a diploid nucleus within a cell. It creates, through meiosis, we're going to make 4 haploid cells, but it's going to undergo 1 round of mitosis. 1 round of mitosis, so each ascus ends up with actually 8 haploid cells that become what we call ascospores. So each of these finger-like structures has characteristically 8 ascospores. 1, 2, 3, 4, 5, 6, 7, 8. So sometimes they're a little hard to see, but that is, if we ask you how many ascospores within an ascus, say 8, okay? Because that's the right number. Some are being, in pretty much any slide you're going to look at, we can't, you know, the researcher who made it can't necessarily control whether some of these spores have been released, but that's how many there should be. So that is asexual reproduction that is sexual reproduction. All right, that's the sexual life cycle. Also, Basidiomycota, this is the sexual life cycle. Basidiomycota, asexual reproduction is not seen very often, so we're not going to worry about that. But in Ascomycota, there are specialized structures that are associated with asexual reproduction. And these structures are known as kinetiophores. So the hyphae can send up these little, they look kind of like little brooms to me. These little broom-like, like little whisk broom structures called kinetiophores. And at the tips of those kinetiophores, we are making asexual spores called conidia. So same deal as asexual spores I've talked about in previous groups. They're great for dispersal. They make them all the time, but they're not a result of sexual recombination. So there's that downside. We also looked at some lichens, which are a mutualistic symbiotic relationship. So symbiotic means you live together, and in fact, this is what we call an endosymbiotic relationship, where one organism is actually living inside the other organism. It is mutualistic. That means it's a plus-plus. They both benefit. So we looked at a couple things. We looked at a slide that actually did a section through a lichen, so we could see both partners. And one partner is the fungus. My pen doesn't want to work. The fungus. Okay, and all these blue threads, that is part of the hyphae of the fungus. The other partner is an algae, or sometimes a cyanobacteria. That is what, this is algae in this case, and I believe, and that is what these dots are. So what does each organism get out of it? Well, the fungus benefits by having the algae live within it because the algae is photosynthetic and can create sugars through photosynthesis. So that is awesome. The fungus gets to take advantage of that. What does the algae get in return? Well, it gets a nice, safe place to live within the tissues of the fungus. It is protected. Also, the fungus is awesome at absorbing water and nutrients, right? That was one of the main important things of the growth form of the fungus, is that it has a really high amount of surface area, which makes it really, really good at absorbing things through its body wall. So it can provide this algae with protection, with water, and with nutrients. We also looked at the 3 growth forms of the fungus, known as crustose, foliose, and fruticose. So the crustose lichens, finding a good one here. It's a really small one, but a crustose lichen, think of it as being encrusted on, right? It looks like you would have to use your thumbnail or something to scrape it off. It grows very, very tightly to its substrate. A foliose lichen is sort of the more loby, leafy looking one. It does rise up above the substrate a little bit. It looks like you kind of pick it off pretty easily if you want, but it's certainly not growing up high. A fruticose lichen is a shrubby lichen that kind of grows, might actually kind of grow up in a branched form. Sometimes it even has little structures that almost kind of look like fruits. They're not, but kind of looks that way. So they could be standing upright and shrubby, or a lot of fruticose lichens grow downward like this. So think of something like old man's beard you might have seen hanging from trees. Okay, I know this is a lot of text. So let's talk about plants. So we looked at a number of different plants, both non-vascular plants, seedless vascular plants, and seed plants. For all the plant labs, you need to know the kingdom and phylum of each specimen. Well, the kingdom's easy, right? Because all plants are in the same kingdom. It's called Plantae, just the Latinized version of the word plant. But you also need to know the phyla of each specimen. You'll also need to answer questions about alternations of generations, which is part of the sexual reproductive cycle. So all plants do this alternation of generations, but it looks a little different depending on which group you're looking at. And one of the really important things that you'll need to be able to do is to identify the sporophyte versus the gametophyte generation for every plant phyla, and its ploidy, meaning how many chromosome sets does it have? Is it haploid or is it diploid, right? The gametophyte generation is always haploid. The sporophyte generation is always diploid. So any of the structures or specimens we looked at in lab, you might get, put in front of you and asked, hey, are you looking at the sporophyte or the gametophyte generation? So when we're into the angiosperms, you need to identify them as either being monocots or eudicots based on either their root, their leaf venation, or their floral parts. Also, we looked at several slides of roots and stems. You need to be able to identify a slide as being a root, a stem, or a leaf, and know whether that organ belongs to a monocot or a eudicot. Also, in this lab, we're going to have several models, okay? And you will have to identify that model as a root, a stem, or a leaf, and know, again, if it belongs to a monocot or a eudicot. Okay, phylum Heptaphyta, the liverworts, the lovely, lovely liverworts. Okay, let me just grab my notes here. So we looked at what's called a thalloid liverwort in this lab, and particularly its genus was Marchantia, TIA. And in this genus Marchantia, we have we have dioecious gametophytes. So this kind of leafy part, this is the gametophyte, it's what we call the thallus. But also part of the gametophyte tissue are these gametophores growing out of it. And we have separate male and female gametophores. The ones that have this kind of scalloped edge are the male. The ones that look a little bit like, I think they look kind of like palm trees, those are the female. So the gametophyte generation is dominant in all the non-vascular plants. And the phylum Heptaphyta is a non-vascular plant, meaning it doesn't have xylem and phloem. So let's look a little closer at those sexual structures. So if I were to zoom in on one of those female gametophores, which is known as an archegoniophore, and if I looked underneath, so if I look kind of underneath one of these sort of branches here, for lack of a better term, I would find archegonia. It's an archegonium. At the base of this archegonium is the egg. This egg doesn't go anywhere. It's stationary, it stays put. If I were to look up close at the male archegoniophores, known as anthoridiophores, I would find these club-shaped structures called antheridia. Within this antheridia, we are producing numerous sperm. These sperm will be released to the environment. They will need water to swim through so that they can make their way to the egg to fertilize it. Once fertilization has occurred, the zygote will develop right within the tissues of the maternal tissues of the gametophyte, and the sporophyte is going to grow directly out of it. The mosses are phylum Bryophyta. They are also a non-vascular plant, so their gametophyte generation is dominant. That is the gametophyte generation. The sporophyte generation is this stalk with the capsule on it that grows out of the gametophyte, and their life cycle is very similar to the liverwort in that we have Archegonia with non-motile eggs and Antheridia with motile sperm that will swim through water to find their way to an egg, and then the zygote and resulting sporophyte generation grow directly out of the gametophyte. So let's look at this a little closer. So let's imagine we've zoomed in to the male structures on a moss. They look pretty similar to the liverworts, but I should say we're going to find them at the tips of these branches of the gametophyte. So the male structures, again, are called Antheridia. They look very similar, these little club-shaped structures in which numerous sperm will be produced. In the female, if we're looking at a female structure, Archegonia, kind of a little harder to see. So there's an Archegonia, there's an Archegonia, and we have an egg in the base. The sperm will swim, so water is important here, and make its way to the egg where it will fuse in fertilization. A diploid zygote results, and then through mitosis it becomes an embryo and then starts to become a maturing sporophyte, which will grow directly out of the gametophyte. The sporophyte includes a capsule at the top. This is a long section through that capsule. Within that capsule, specialized spores called sporocytes will undergo meiosis to produce haploid spores. And that is what you are seeing right here. Spores. Okay, so let's talk SVPs. SVP stands for seedless vascular plant. These are plants that don't have seeds, so we're not into the seed yet, but they do have vascular tissue, aka xylem and phloem, allowing them to conduct water and nutrients from the ground and allowing them to move their photosynthates, the products of photosynthesis, around their tissues wherever it's needed. So that's a pretty big deal. It allows the seedless vascular plants to get a lot bigger. So let's look at a couple phyla within the seedless vascular plants. Actually, we're just going to look at, no, we are looking at a couple phyla. So we'll start with Monilophyta, ferns, and horsetails. And what you're looking at right now are structures of a fern. So we call the leaves of a fern, we call those fronds. And the sporophyte is dominant. So that's another big thing that's going to be happening now, is that instead of the gametophyte being dominant, the sporophyte generation is going to be dominant. So that's sporophyte. It's going to be the large, conspicuous plant that you see. The gametophyte is greatly reduced. It is green. It has some photosynthetic capability. It can live independently. It has little structures called rhizoids that will help anchor it to the whatever substrate it's on, usually the ground. And so as the gametophyte, again, this is haploid. Okay, that's a haploid structure, one set of chromosomes. So in the ferns, the gametophyte is bisexual, meaning that on this one structure, we have both Archegonia producing egg and Antheridia producing sperm. Again, like we've seen in other groups, these sperm are flagellated and they need a drop of water or some amount of moisture in order to swim to the Archegonia and make their way to the egg. And then the sporophyte will grow directly out of the gametophyte, as in other, that's a really awful drawing, but as in other plant groups. But in this case, the sporophyte's going to get big and very quickly will overtake this gametophyte and be completely independent. Now, let's talk about the spore-producing structures, because those are going to be a little different in our ferns. So our ferns are going to have what we call sporophylls. which basically phyll means leaf and sporo means spore. So it's going to have leaves that will bear spores. And these, oh, I don't have a picture that I want. So basically, if you look at the back of a fern leaf, if it's a fertile sporophyll, there will be these little, sort of rusty colored dots that we call sori. Here is an up close of one of these sori. Each of these sori has numerous sporangia. So there's a sporangia, or sporangium, singular. Within this sporangium, we are making spores. Okay, so take a second and think about this. So we have many spores being produced in each of these sporangia, these little stalked sporangia. And then if we look at this sorus as a whole, there are numerous sporangia. And if we were to look at a leaf, a fern leaf, like you did in lab, again, this is not a really good example of a fern leaf, they look different, but each leaf would have numerous sori, right? So there's a sori with a sporangia with lots of spores. Also within the phylum Monilophyta are the horsetails. Okay, so the horsetails have a hollow stem. They have greatly reduced megaphylls. So I know you look at this and you think, that's a microphyll, right? No, it's actually a megaphyll. And remember, the distinction is a microphyll has one strand of vascular tissue, whereas a megaphyll has more than one strand of vascular tissue. It's just greatly reduced in this case. Not seen here, but many horsetails also have whorled branches coming out at the nodes. In the horsetails, instead of having the sporangia kind of spread out on a big flat leaf, the horsetails obviously don't have a big flat leaf. Their sporangia are clustered at the top in a structure called a strobilus. So basically what you have are numerous sporangia clustered together to form this strobilus. Sometimes this strobilus also gets called a cone. And again, as a seedless vascular plant, you're looking at the sporophyte generation, and the sporophyte generation is dominant. The gametophyte, not shown here, is really, really small in the horsetails, and you would never, ever even notice it. Okay, another phyla of seedless vascular plants that we looked at are the phylum Lycophyta. So the common name here, by the way, is club moss, and sometimes you will need to know the common names. So use this PowerPoint as your guide, right? Because we write the common names on there. Horsetail, fern, club moss, it's all on here. So the Lycophyta have those microphylls, right? So just one strand of vascular tissue. The Lycophyta also have terminal strobili. So we have a number of sporangia that are clustered together. A lot of times there'll be like a little small leaf tissue that we would call the sporophyll underneath the sporangia, but it's clustered together in this terminal structure known as a strobilus. All right, Conurophyta. So we looked at, so now we should say, we're in the seed plants. Okay, so I'm going to end this video here just for sake of a break, but we're going to come back and I'm going to talk about these seed plants.